So we're down to the final two chapters in this course. We're working on chapter 31, which is on the nervous system. And we were introduced to the cells of the nervous system back in chapter 25, because there's really one key kind of cell in the nervous system, and that would be the neuron. And so when we looked at neurons before, we said that they had a cell body, and then they had branching extensions. And we couldn't see a whole lot under the microscope, but there were some schematics that were added to our notes. And so we're building on that we learned in chapter 25. When we take a look at your whole nervous system, it's composed of two different parts. We're going to talk about your central nervous system and your peripheral nervous system. Your central nervous system is basically your brain and spinal cord, whereas your peripheral nervous system is everything else outside of the central nervous system. And so here we would find nerves that were bundles of sensory and motor neurons, so a bunch of these neurons together. And then ganglia, which is really just where all these cell bodies are gathered together. And so we do have ganglia within our nervous system. And really what we're talking about is a collection of the cell bodies of, again, those neurons. We're going to come back to the two divisions of the central nervous system. Before we get too far, we're going to talk about the two different kinds of nerves that you have. In your peripheral nervous system, you have sensory nerves, which are in other resources called affector nerves, which basically go from your extremities to the central nervous system. We have one-way direction in our nerves. So we're going to have nerves in our central nervous system, which basically process that information. So this is the peripheral nervous system to the central nervous system, and then back out. So this goes in, and then this goes back out. So the motor neurons go out from the central nervous system to make your body do what you need to do to respond to things. So motor neurons also have the name effector. You can have a motor response. You can have basically a nerve impulse that escapes the central nervous system, and that's what your reflexes are doing. There's no time for that nerve impulse to get to the central nervous system for processing. It really just relies on the sensory and the motor neurons to do that work. That doesn't need any processing. There is a crash course that I put on the Moodle site. There's also the anatomy physiology ones that take it a little bit further. So as a basic sort of introduction, I'd suggest that 12-minute overview of the crash course. But if you're curious anywhere beyond that, then I would suggest going further. So this is just an image about the sensory input going through the affector neurons, and then this integration in the central nervous system. And then the other motor neurons, motor neurons or effector neurons are going back out to do something. And so again, these ones were all in the peripheral nervous system, where that integration is in the central nervous system. If you have something that bypasses that central nervous system, that would be a reflex. So you might have a reflex if someone, your eyes will close without you thinking about it. If someone throws something at you, the knee jerk, like a doctor will check your reflexes. That's what we're talking about those. So neurons are the specialized cells for carrying signals, electrical signals. They are the functional units of the nervous system. We've talked about them before, how they have that cell body and then those branching extensions. And the extensions are called either dendrites. Those are the ones that come off the cell body. So the branches here off the cell body would be the dendrites. And then there's always one long extension, and that is the axon. And so this would be the axon, where these are the dendrites. So we're going to talk a little bit more about the neuron, because there are a few key components of it. And there's a better diagram on the next page. And really what it's going to illustrate is those components. And also along the axon are little beads, if you will. They're little insulated bits of this electrical impulse transmitting nerve cell that basically have some key jobs to do. So those little beads along, if you will, the beads in my analogy. Oh, I had written it there. They're not really beads, but they're another kind of cell. And so they're technically a cell that's wrapped around the axon, kind of like a jelly roll style. And basically, it also has an insulating material called myelin in it. And so it's sort of interchangeable whether you call these beads Schwann cells or the myelin sheath, because it's technically both of those. And basically what they do is they insulate parts of the axon from the nerve transmission. And so what happens is that the nerve transmission is allowed to pass faster because it only bounces through those exposed bits. Those exposed bits are called the nodes of Ranvier. One of the videos calls them Ranvier. I think Ranvier sounds way fancier. In the crash course, he talks about it as a working title to the next Harry Potter novel. So that's what we're talking about. The nodes of Ranvier is where we have the electrical impulse actually being transmitted along the axon. And so they're significant because they speed up the nerve impulse down the axon. So let's take a look at a better diagram than what I just tried to scribble there. It's got all of the parts labeled on it. And so just as a reminder, we had the cell body, and that's where the nucleus is. The branches coming out of the cell body are called dendrites. And you can see the nucleus in there, right? Down here would be the axon. And so that's that long branching extension. And along the axon, we have these insulated bits, these Schwann cells that contain the myelin sheath. And so over here, you can see them a little bit better. This is a cross section of a Schwann cell. And so it's a Schwann cell with like myelin as the jelly in the jelly roll, if you will. I don't know if that really helps people. Certainly nothing I would write down in the formal notes, but as a memory device, you can kind of imagine it like that. So the key thing about the Schwann cells in the myelin sheath is that it insulates most of the axon, but leaves little pieces of that axon exposed. And those pieces are called the nodes of Ranvier. And it's really important because the nodes of Ranvier is where we're actually going to have the transmission of electrical impulse happening. And that's really what we're talking about when we're talking about nerves and the cells of the nervous system, which are neurons. And so I put in the words here, a coiled Schwann cell is the myelin sheath. It's tricky to see how those words relate to each other. So if you want, you can just think jelly roll style, where the myelin is the jelly of the jelly roll, and the jelly roll cake part is the Schwann cell. We've already talked about this one. So the components of the nervous system would be the central nervous system. So that's the brain and the spinal cord. And it contains fluid-filled spaces. So similar to ventricles in your heart, you have ventricles in your brain. And you also have the central canal of the spinal cord. So we have brain and spinal cord being part of your central nervous system. The peripheral nervous system is everything else. So all of the spinal nerves that are coming off of your spinal cord, and then anything outside of that. And so any of the cranial nerves too, so cranial nerves going off your face or through your face. All of the other nerves other than the brain and the spinal cord are your peripheral nervous system. So we're going to talk more about your peripheral nervous system for a little bit. We're going to talk about how a nerve impulse is transmitted. Then in the second video, I'll talk about synapses, and then also a little bit about some brain basics. Okay, so the two parts of the peripheral nervous system, if you can kind of follow the flow chart down, we had the central nervous system and the peripheral nervous system. The peripheral nervous system is divided into two parts. So we could have the somatic nervous system, which is all your voluntary thoughts and actions, and the autonomic nervous system, which is all your involuntary stuff. When we talk about, the fight or flight mechanisms, fight, or the rest and digest, those are all part of your automatic or autonomic nervous system. The somatic nervous system, or those voluntary, are all the things that control your voluntary muscles, your skeletal muscles, your striated muscles, and mainly respond to external stimuli. So as we talk about your autonomic nervous system and these two components of it that I've already given you kind of the common sort of memory devices for, all of these are going to control your smooth muscles, your cardiac muscles, because you remember they were involuntary, voluntary, and really are included. This whole nervous system controls several organs of various body systems. So we could think about your intestines, we could think about your diaphragm, your whole circulatory system. Certainly there's a lot that happens without you needing to be conscious of it. So here we have that sort of branching. I really love the pictures of this. So we had, your nervous system generally can branch into your central nervous system or your peripheral nervous system. So now in the notes, we're branching your peripheral nervous system into your voluntary or motor neuron system, nervous system, or your involuntary nervous system, which is called your autonomic. And I think automatic, but it's autonomic as a word. And so this whole idea that I said, the rest and digest or the fight or flight, those are the kind of the catchy names for the two divisions of, or two of the divisions of your autonomic or your automatic nervous system. And so your fight or flight is sympathetic to your needs. It is the sympathetic division, and it really carries nerve signals that prepare your body to fight or flight, and use up a lot of energy. In sort of contrast to that, or usually in balance to that, we have the parasympathetic nervous system, which is like the chill out system, the opposite of the fight or flight, which is the rest and digest. And it basically does the opposite in that it conserves energy. And so we have these two different components of our automatic nervous system that help maintain homeostasis. We also have the enteric division, which basically is your gut brain. If you've heard that, they had a really good series on CBC, I think it was last summer, about the gut brain. That idea, that's the enteric division, and we're not really covering that all that much, but you can see down here all the different systems that are involved in your autonomic nervous system. And it's always a balance or a bit of a tug of a war between the sympathetic and the parasympathetic division of that autonomic nervous system. The actions of the sympathetic and the parasympathetic neurons are basically opposing, and they really do help regulate the internal environment. So it's usually, like I say, this balance, and it shifts as need be, for your body. So that parasympathetic is the rest and digest, or resting or fueling, whereas the sympathetic is the fight or flight response. And basically, these systems work on the exact same targets in the body, but on one target, it's going to decrease activity, and the other is going to increase activity. So in this way, like I say, it's that tug of war, it's that balancing effect of the two actions that helps maintain homeostasis. It might be easiest to see in this diagram, where basically we have that sympathetic, the fight or flight, or the rest and digest, where we do have, the actions here being opposing. And so in the rest and digest, your pupils are going to constrict. If you're getting ready to fight off the bear, your pupils are going to dilate or get bigger, right? So one gets smaller, one gets bigger. In the rest and digest, you're going to drool, I guess, or stimulate saliva production, because that's part of the digestion, right? In the fight or flight, you're going to get dry mouth when you have to give that speech in front of the class, right? That's that sympathetic division of your autonomic nervous system. And basically, you go down the line here, all of these targets in the body are affected oppositely by these two different parts of the autonomic nervous system. And generally, like I say, it's usually in balance, unless need be otherwise. The next part of the notes here is kind of that detailed bit. I used to joke about it being the 5-mark question on the exams. Well, we're working things a little bit differently right now. But in these ones, we're going to talk about nerve signals and their transmission. There are two quite good videos that I've put, or several quite good videos that I put on the Moodle site that give you this information in various ways. And so basically, we're going to talk about how there is a change in charge across the plasma membrane of the neuron. And so we're going to talk about the energy or the membrane potential or the potential energy difference from one side of the neuron to the other side of the neuron. And what I'm talking about is not one end of the neuron to the other end of the neuron, but one side of the membrane to the other side of the membrane. And so if I were to draw one little node of Ranvier here, so that would be a Schwann cell, this would be a Schwann cell, and I've got that exposed bit of axon, that's really where we're centering this next part of the notes and our discussion. So if I talk about membrane potential or energy potential, there is a difference in charge across the membrane. So from inside to outside, there is a difference of charge. And so we say that it is negative charge, but really what we're saying is that it's less positive. So when a neuron is resting, we say that it is negatively charged. Well, it's not. We have positive ions out here and positive ions in here, but we have more positive ions outside than we do inside. And so that's why we say that it is negative. So it says, you probably answered that in your pre-reading a little bit here. Let's get into the details of why it is negative. So a resting potential is the difference in charge from inside to outside of the neuron when it's not firing. And so the resting potential exists because there's a difference in ion concentration inside and outside the cell. And so the details are listed here. The way I remember this is, you ready for this? It's a terrible memory device. Na, na, na. I'm going to switch colors here just for fun. Ka, Ka. And what I'm talking about are the ions, the potassium ions and the sodium ions. And now that's not the whole story here, but that gives me a place to start my understanding. So we're talking about the potential difference or the difference in charge of a resting neuron from outside and inside the cell when we compare this. So outside the cell, we have a low amount of potassium ions and a high amount of sodium ions. And so basically, there are still potassium ions outside the cell, but there's just not as many as inside the cell. And this one says inside the cell, we have a lot more potassium ions and a lot fewer sodium ions. And so if I were to complete my diagram here, not just na 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 ka ka, but we'd also have some sodium ions inside the cell. And so if we take a look at this, we have an overall more positive outside the cell and less positive inside the cell. So that's what we're saying. There is a difference in charge. And so we say that the cell has a negative potential difference. Well, it's not anything about it that has a negative charge. It's just less positive than outside the cell. So I'm going to keep going with that because really understanding this resting potential is key. It's the starting point for a nerve impulse or for a nerve action potential. And so how it gets like this, because there is a concentration gradient, we've talked about active transport, and an example of that was the sodium-potassium pump. It takes energy to maintain these concentration gradients. It would take energy to pump sodium from inside to outside, or it would take energy to pump that potassium from outside to inside. And basically that's what the sodium-potassium pump does. It moves both of those ions in opposite directions against their concentration gradient. And it uses energy in order to do that. So that's going to come back into our understanding of how a nerve fires. But that's really just the starting point, that resting potential, and how we could talk about a negative potential difference for a neuron. So here we have a simplified, or well, simplistic somewhat drawing. And so here, this is the way your textbook did it. And I've always had trouble with this, where it looks like there are negatively charged things, but we've already said it's not actually like that. There's just more positives outside and less positive inside. If we were to talk about that na 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 ka ka, take a look at how much sodium is outside the cell. Well, there's still sodium inside the cell, but quite a bit less. And oppositely, the ka part of my memory device there was that there was mostly potassium inside. Well, there's still some potassium outside, but we do have that concentration gradient. And so we've got two different concentration gradients, and we've got this overall more positive and in comparison more negative, even though there's nothing that actually has a negative charge. I think the schematic put the negatives on those proteins just to try and help you conceptualize it, but I do take a little bit of issue with that because that's not really what we're talking about. The last part of this image is that sodium potassium pump. It uses ATP to maintain those concentration gradients because that's how a resting cell is set up, outside and inside the neuron. Okay, so now that we understand the resting potential a little bit different, um, we could then talk about, well, what happens when a nerve is stimulated? And so basically, as a nerve is stimulated, there's a nerve signal that's going to begin as a change in those relative charges. And so we're going to talk about how this electrical charge changes as a nerve fires. And so it's going to start with a stimulus that alters the permeability of one section of the membrane, allowing ions to pass through. You remember we had a potential, or so we had a concentration gradient set up. And so basically, if we open up the membrane to ions passing through, we're going to have ions move, right? Through passive transport. All of this is going to change the charge. It's going to change the voltage of the cell. If we get to a point called the threshold, which is a specific number as far as the change goes, that potential difference or voltage as we talk about it, then the energy or the nerve is going to fire. And what we talk about with a fire is not a resting potential, but an action potential. That is a nerve impulse. Okay, so an action potential is again going to change this membrane voltage or potential difference that we've been talking from the resting potential up to a spiked maximum level and then back to the resting potential again. So I really like the next slide because it has a graph in the center that I find really quite useful. And so I'm going to just zoom in on the graph here for a minute. And so this graph is on the side is the voltage, if you will, or the potential difference. And so if you've taken physics before, potential difference is measured in voltage, just like batteries are measured in voltage. So a battery has a potential difference from one end to the other. And so maybe this is a 9-volt battery. So same as that, we're talking about a change in charge, only this time it's much, much smaller because we're talking about millivolts. And so if we take a look back to this vertical axis of our chart here, we have the resting potential down here. Remember, I said that the cell is less positively charged, or in another word, negatively charged. It's down here at -70, not volts, but millivolts. And so that's what we were talking about with that na 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 ka ka. That's our starting point. That's our resting potential. And the rest of this, as we travel through time, illustrates the key components of an action potential. So we have a stimulus right there. So that would be the stimulus. And that's going to alter the permeability of the membrane up to a threshold level, like the last note said. If we get to that threshold level, then we're going to have a rapid change in the potential difference up past where the charges are equal to where the neuron is actually more positively charged than outside, and then a return back down to the original. And so I see this as a few different components. So we have the resting potential is 1. That stimulus to the threshold is 2. The upward spike is 3. The downward return as 4. The little dip and then the return to normal. So I'm just going to talk through those 5 points as we move through this slide. So the first thing we've talked quite a bit about, and that resting state. And so here we have lots of sodiums outside and lots of potassiums inside. And so this is the resting state. We have voltage gated channels are closed, and the resting potential is achieved at negative 70 millivolts. A stimulus would be this number 2 section of my notes here. So a stimulus is going to open some sodium channels and allow the sodium to pass from outside the cell to inside, because that was the direction we had the concentration gradient set up. This is going to change the charge of the neuron. It's going to make it more positive as those positive ions move into it. As it increases, it could hit a threshold if enough of those sodium ions come in. So as it hits that threshold, that opens up more sodium channels and more sodium come in to the point where, like it says on the graph here, the neuron is now more positive inside than it was outside. Right? We still have all the potassium inside, and now we've got a whole lot more sodium inside too. So that would go for this increasing part of the graph. At the top of the graph, things change. We're going to have our sodium close. So everything on this side of the chart is about sodium, and everything on this side is all about potassium. So the sodium channels close and the potassium channels open. Well, remember, we still had lots of potassium inside the cell. And so with that concentration gradient, that's going to allow the potassium to go outside the cell. This is going to again make the cell inside less positively charged. And so we see this return in the potential difference as we compare the inside to the outside of the cell. So number 5 is a bit of a dip. It's a bit of an oops, if you will, in that the potassium channels are just a little slow in closing, and so there is a bit of a brief undershoot. And here you can see, because of the concentrations, we're back to that negative 70 millivolt. Now, I have a bit of a problem here, because if you're keeping track of the ions from one side of the neuron to the other, we've got a problem. We've got to get it back to the original, because at this point we'd have a lot of potassium outside and a little some sodium inside. This is still more positive outside and negative inside, right? But we need to return by use of the sodium potassium pump. And so we need energy to think. We need energy for another action potential to happen, which is probably why that just made me tired. So this idea about action potentials, as we've gone through kind of the 5 points of it, and like I said, there's a few videos on that, so if my words didn't make sense to you, then go ahead and check out the other animations and narrations there. But the action potential basically propagates itself along the neuron. So remember, we had these action potentials. They're in a one-way chain, and so we have nerve body and axon, we're going to have only nerve impulses traveling in that direction. It's a one-way street. You remember that we had 2 different kinds of neurons, one going into the central nervous system and one kind coming out. That's because of this one-way street that neurons are. So the action potentials are always propagated from cell body to synapse. This is the synapse down here. I think I forgot to label that on the last time we saw that. So the synapse, and at the synapse, we'd have another neuron interfacing there. They're all or nothing events. So it's not like if you touch the hot stove, you're going to have a bigger action potential. You're just going to have more action potentials happen. And so the strength of the stimulus changes the frequency of the action potentials, not the strength of the action potentials. This last little bit here, before we get into the synapse, is trying to just illustrate that. And so what we would have would be a tiny part of our axon being exposed, and it basically travels down. The action potential travels down like a wave, it says. And so it moves down the length of the neuron through these nodes of Ranvier, the exposed bits of axon here, in this kind of wave-like action. And so one action potential here is going to stimulate the action potential down which is the stimulus for the next action potential, and like it says, down that exposed bit of axon like a wave. If you remember, I did say that these were insulated because it allows for that nerve impulse to jump from node of Ranvier to node of Ranvier. If we have an action potential here, that is the stimulus for the next part of the exposed neuron. So that's that saltatory conduction or that jumping type action.